That would give any.
I didn't have high blood pressure before. I was probably going to get it, but crap, looks like now I have it.
Well, my friends from internal medicine residency, he's been on losartan, I think, since he was 23.
Whoa.
It's just wild.
Yeah, I'm like hoping if I eat healthier and I lose some weight, I can defer medicines a little bit longer.
Yeah.
Right here. Yeah, exactly.
Thank you. Have you been in service recently?
I was on Thanksgiving.
Oh. Is that a good time to be on or a bad time to be on?
It's a mix. You have to make a choice of like holiday travel if you want to do anything, because you obviously can't. But I actually sometimes find it helpful to do clinical work during like break times. So then grad students, postdoc, like don't call me. Just go
Totally.
see your family or do whatever it is you do, and then
Get out of here.
don't call me. I'm not going to answer anyway.
Yeah, I usually volunteer aggressively to do the heart failure service over Christmas. It's usually pretty light.
Yeah, patients are more eager to get out of there.
Yeah. Yeah, it's great.
That's good.
Heart failure service got down to 5 patients.
Oh, really?
Bananas. It was 14 when I left, but.
That was irritating. I've done like multiple Christmases in the past 5 years. I think it's actually the first time we didn't do Christmas for many years. Or some.
I find most people pick a holiday they like, either Thanksgiving or Christmas or New Year's, and they work. Thank you so much. They work one of those.
All right. Do you have any video or audio?
No.
Um,
Making me feel bad. No video or audio.
yeah, but just we better test
Do
it.
you have any AI generated content?
No. No.
But could you prove or disprove that?
As
whether
good
it's
as that
what you
seemed to
said?
go last time. I love that introduction. I made this with AI. It didn't go well.
Yeah, as you can see, it's pretty bad.
And at first I thought he was just being modest, then I was like, oh yeah, it is. The pictures they don't say words.
No.
So that picture, if you Google for Graham Walker, that picture shows up. You know, like I Googled for him like 2-3 days before his talk. Like, wait a second, I thought he looked different. And then in the talk, I was like, okay, that was just a fake Graham Walker. And then he eventually did look like what I remembered him as.
Oh man.
But that picture has now made it tagged as Graham on the internet and be there forever.
I noticed recently that you want to make an example of the plot and you ask ChatGPT or whatever to make that plot for you as an image. Terrible. But if it makes that plot for you as code, very good.
All right.
It's very interesting.
Yeah, I prototype all of my visualizations now in ChatGPT.
Yeah.
You don't need to
Great.
even real data.
By the way, we're being recorded right now, so just
Oh.
FYI. I can stop that if you wanted to say something.
That's okay. I am all positive.
Is this an introduction to the community? Sort of, but you also did interview / job talk kind of thing here.
Yeah, but I think a lot of
Was
people
that like
probably
2
didn't.
years ago?
Yeah, it was. It's a little while ago. There'll actually be some of that in there, but yeah, no, it's
Was that even in person?
it was, there were, well, no, the job talk was not in person, the chalk was.
Sure. Don't have a visual memory of you standing up.
No, I did not do that.
Must have presented here before, but I don't have this visualization of you standing here.
It was a separate job talk, no colloquium. So it is a little bit of an introduction. I kind of anticipated it before.
We're fire now in about 5 minutes from off.
I know, I was like, feels kind of quiet, but then of course I realized that I always get here at exactly 12:01,
Yeah,
so.
why would you show up 2 minutes early?
The prior meeting goes till 12, so.
That a line for food is probably forming right about now.
Right, Jonathan, just FYI, I'm getting drafted by Sean and Mark to
Oh, yeah,
basically compile a list.
Mhm.
Because apparently what we are doing in the Department of Medicine is, and I was talking to the dean's office people,
Mhm.
like their expected process is different.
Who's expecting what? I mean.
Exactly, so I'm basically sending point messages one-on-one, it's like, if this was the case, how should I do it? And then I get a process, so what needs to happen is, as part of the SOM committee, you're on it too, right? Yeah, you are.
Yeah,
So we take candidates to UN,
right.
directly chair, to confirm,
Mhm.
notify Kurt and Natalie
Mhm.
that medicine is interested, thumbs up. Only then it will show up in that in that dashboard for the search. Okay, fine, guys.
I'm just trying to help people.
And so what I'm gonna do is, I'm confirming with Sean, and I'm just gonna take your entire list and whatever Lenny suggested,
Mhm.
and just be greedy and say, this full list, medicine, thumbs up.
Yeah, at least for Zoom.
Exactly, at least for Zoom, because there's no much downside.
It was more, yeah, there was like, people, there have been people sitting in this applicant pool for a
Mhm.
long time. They're gonna move on.
Mhm.
if you Don't act. It's like, what is Sean doing here versus if we have to just go back to you and anyway? It was like, Sean's like, hey, I asked Sean, like, Sean, can I do this? Do you want me to do this? Sean's like, yeah, go do that, Sean. I'm like, okay, Sean said so, I guess he's the one directing this.
No, he's not, so talk to him too. He's the chair of the A&P committee. In fact, the more he gets involved in this,
More problematic.
the more problematic later, because he's the chair of the A&P committee.
Yeah.
But he told it himself, so we're gonna talk on the phone today and sort out. Because the last thing you want is getting held up because of some bureaucratic snafu, and provost says this person shouldn't be on.
Yeah, I'm looking forward to this.
Great.
So long await.
Oh,
Building.
Had it marked on your calendar for a year before it was done.
Years in preparation. We made it. Maybe give it like 60 more seconds, we'll start the.
I know, it's like, it's very hit or miss actually, in this area. If you have been down to like 3 months, I would say like, it's better. I would say it's better, on average it's better. So if you're down there, I think you're more likely to find good players.
Tina, are you on the call today this afternoon? 1 o'clock, some committee with data and clinical in it.
Maybe. I don't know. What is my schedule? Is it Sylvia's thing or is it Ruth's thing?
Ruth and Sylvia.
Probably. The data, yeah, research.
Yeah, something. Clinical something.
In person?
I'm not going in person, go here.
Yeah, yeah.
Do you see the briefing slides?
No. I haven't.
I looked at it, I can't make head or tails of it, like, what is this?
I went to the.
People outside. So the one on Tuesday?
Yeah.
The one where you are now present as the CTSA informatics?
Well, it was kind of fun. So,
Are
Kurt,
you gonna be
very much.
pressured for time or plenty of time?
I would say on the pressure side, I got through in about 50 and then I cut some stuff, so probably 45.
All right, fantastic, everybody. We're gonna just formally get started because we're very privileged and very glad. I feel lucky we have, it turned out after 7+ years, I'm not the most junior faculty in here. Dr. Dan Katz, we are very glad has finally joined our division. It's been all sorts of exciting he's been working on, but also just glad to have another physician in the group. Dan, please just tell us about yourself in general, and he seems like he was prepared for this.
Yes, well, I've been coming to the Colloquium enough that I should know what to expect. So this is my disclosure slide, and I'm going to be showing some prelim data today, which I'm excited to do, but I'm supposed to disclose that it's preliminary. Share it wider. We're excited about it. So I was going to talk a little bit about my path because I know Jonathan asked everybody, so I will tell you a little bit. So I grew up in Chicago and went to undergraduate medical school at Northwestern, and really Northwestern is where I got first interested in working with biomedical data and worked very closely with Sanjeev Shah, who's there, who still is a collaborator. And that's really where I got my start. I went off to Boston. I did my internal medicine residency and my fellowship there in cardiovascular medicine. And so after spending 7 years in Boston, that's where I really got interested more in multi-omic data, which is the subject of my talk today, working with Robert Gerson and a number of other great collaborators who I still work with
So
now.
quick question.
Yeah.
Were you involved in that work, classifying heart failure with preserved ejection fraction into 3 subtypes?
Can I get to the next slide? Yes, I'll show you.
That is awesome. They talk about it all the time now.
Yeah. Yes, it remains my highest cited paper. So yes, I'll definitely show that soon. And then I got to Stanford in 2021, where I did my advanced heart failure and transplant cardiology fellowship and joined the faculty the year after as an instructor doing working in the advanced heart failure and transplant cardiology group. So I take care of people who have had transplants or LVAD devices put in to treat their heart failure. I also take care of heart failure patients before they have the need for those serious surgeries. I don't do surgery. A lot of people don't know that, but I don't do surgery. I send people to surgery and then take care of them afterwards. Of course, I joined the faculty formally here in September as an assistant professor. Actually, I'm joined between here and CV Medicine, and I'm very excited about the prospect of really getting more cardiovascular data into the work that we do here, and that's really a main part of my role. So I'm going to split this talk up into three and a half sections. So I'm going to talk a little bit about multi-omic observational data in cardiovascular health and disease. I'm talking about omics in clinical trials and experiments. And I'm going to talk about running a bioinformatics center. And then I'm going to hopefully stimulate everyone to talk a little bit about the future of multi-omic data in both bioinformatic and biomedical informatic environments. And my goal is to describe how multi-omic data is increasingly part of biomedical research and to invoke a vision of the future for multi-omic. So let's start with observational data. So I'll start off with a pair of heart failure patients that I saw on the same day in my clinic that illustrate why I'm so drawn to this type of research. So on the left here is Mr. Jackson, on the right is Mr. Ahrens. They're of similar age. They have similar left ventricular dysfunction in their heart. And they're experiencing vastly different symptoms of disease, of congestive heart failure, of shortness of breath, which is really the key symptom of heart failure. Mr. Jackson, he's riding his bike regularly, and Mr. Ahrens gets short of breath just while walking. And so it's fascinated me why, despite having similar looking hearts on echocardiography, why these two patients are having very different lives. And I really, became fascinated with this idea that multiple combinations of risk factors, etiologies, then combine together to form these clinical syndromes. Heart failure is an example, but substitute your favorite disease in there. They are all heterogeneous. And that results in very heterogeneous clinical presentations and trajectories. And so, yes, this is that study. So this is how I got started in this area, working with Sanjeev, is that we took a pile of really what you would call phenomic data. So all the phenotypes that we could gather in a clinical setting. So lab results, echo, EKG, anything that we could measure clinically, and we put all that data into a hierarchical clustering algorithm and looked for sort of an idealized number of groups. And we showed that if you could use this phenomic data to subcategorize people in an unsupervised way, you actually broke them into 3 classes of individuals that had very different experiences clinically. Those in phenogroup 1 did well over time, and those in phenogroup 3 did substantially worse. And it was on top of existing risk scores. So something about their more global data collection was allowing us to get insight into their disease. So I really felt like if phenomic data was good, then multi-omic biological data has got to be better, because we're getting much closer then to the actual phenotype at the molecular level. And so we're collecting an ever-growing amount of this data. And particularly, we're doing it across very diverse populations. That's an important point, because multi-omic data can vary a lot by environment, but it can also change by genetic ancestry background and lived experience. And so it's really important to generate this kind of multi-omic data in a lot of individuals. So before we jump in, I think we should say exactly what I mean by multi-omics. So to me, multi-omics is the comprehensive collection of biomolecular measurements across at least 2 omic categories. We know that the central dogma of biology is that genomic data is transcribed to RNA, translated to protein, which then act in the biomolecular milieu to produce phenotype. Multi-omics seeks to understand this interplay in a data-driven manner by gathering data on as many features in the individual omes as possible and studying how they behave. So for example, we might collect measurements of the whole genome, 20,000 gene transcripts, 5,000 proteins, 2,000 metabolites. To me, this is multi-omics. So let's go to the land of the volcanoes. Single omic studies. These are great. They still have an important role, and I'll explain why I call this the land of the volcanoes. But a little bit of background. So a lot of this data comes from TOPMed, which is Trans-Omics for precision medicine. A lot of the cardiovascular cohorts, including the Jackson Heart study, are included in these data sets. They collect echocardiographic data. In this case, we'll be talking about proteomic data, and these are then merged with whole genome sequencing in TOPMed. So this was a very early analysis that we did, where we were looking in the Jackson Heart study at proteomics. So we measured 1,300 proteins, and we looked at the association with left ventricular mass, so the size and thickness of the left ventricle, which is an important marker of future disease. And this is a volcano plot. So this is why I call this the land of the volcanoes, because in single omic studies, you see these kinds of plots a lot. And that's because they're actually pretty useful. So you can see on the x-axis, you've got the beta coefficient of association with LV mass. On the y-axis, you have the negative log of the p-value, and it really helps to just sort of resolve out all of these proteins. So you can see things that jump to the top, like leukotriene A4 hydrolase, which was a protein that we found, cross-sectionally at least, was associated with increased left ventricular mass in the Jackson Heart study. When we looked at incident heart failure, which is the graph on the right, and we looked at the hazard ratio for heart failure, a very different set of proteins emerged. In fact, even though LV mass and heart failure are linked, the proteomics underlying them were completely different. And so we thought that that was particularly interesting in this study. So single omic studies are great, but we want to layer in other omics. We've got multi-omic data. Let's try to use it. So this is called a PQTL study. So in a PQTL study, what we're trying to do is we're trying to link genetic variation to the full proteome. So you measure 1,300 or 5,000 proteins, and then you take the entire genome and you try to look for all the variants that are associated with all of those proteins. Computationally intensive and requires its own GWAS pipeline to be specifically designed for this, because if you're going to try to do 1,000, 2,000, 10,000 GWASs, you're going to need to have something that runs a little faster than say an hour for a single GWAS. And so we built this pipeline with our colleagues at the Broad, which allowed us to do this across multiple proteins. And when you do this across 1,301 proteins in the Jackson Heart study, we identified 569 PQTLs. And this figure summarizes these findings. So for each association between a gene location and a protein, a point is graphed by the position of the gene and the position of the gene that actually encodes the protein. So a cis PQTL lines up along that identity line. That's because the protein is encoded from the same place as the variant. And all the trans PQTLs, which are offsite, so in other words, the gene codes for something else but seems to influence a different protein, those are off axis. I think the real key take home here is that there are just a lot of associations across this that we found a lot of, we basically described the genetic architecture of the circulating plasma proteome. And so what is this good for? So let's take an example here. This is hemopexin. So we found, so hemopexin binds free heme, that's its role in the body, and it may have an anti-inflammatory role. And we found a variant that was unique to African ancestry that we identified in the Jackson Heart study at about 5%, but it's not in European ancestry. So it hasn't been well studied because it's, Black individuals are not well represented in existing GWAS. And so we found multiple proteins that associated with this variant, and a lot of them were inflammatory proteins, suggesting that this could verify the role of hemopexin as an anti-inflammatory protein. But there were no other known GWAS associations. It wasn't associated with disease. So this is really the first time that we were able to learn about it because we were using diverse populations. So then, of course, you want to layer in additional omes. Oh yeah, go ahead.
So where'd you get the data? You collected that data yourself because you can't use the human genome for that, right? Because for your Black patients who have that, what data were you using?
They were sequenced. So all of those
You
individuals,
sequenced them?
we didn't personally sequence them. They were done as part of Trans-Omics. So in part of the TOPMed program, all of those cohorts got whole genome sequencing to create a database for this. Yeah, good question. So you want to layer in other omes. So now we brought in the metabolome. So these were measurements that we did make ourselves in the lab, the metabolome and proteomic data. And so you want to try to get these together, and we use a technique called Mendelian randomization, where we look for shared genetic architecture to connect the variants that we see in the genome to the variation that we see in the proteome and the metabolome. But by doing Mendelian randomization, you're actually connecting proteins and metabolites into likely causal pathways, which is something you can't necessarily do when you just look at the correlational data between those two omes. And so by doing this, we actually identified 224 protein to metabolite causal associations. And when you bring this over to a model, you bring it over to a knockout mouse, it actually verified for 3 of those genes that were knocked out in these mice. We found that 50% of the associations that we predicted based on Mendelian randomization were actually seen in these mice knockouts. And so it shows that the Mendelian randomization approach can really help you to identify likely causal pathways that then bear out in biological systems. Okay, so that's a little bit of observational data, but let's move into omics in clinical trials and experiments. This is where we're going to talk about MoTrPAC. So what is MoTrPAC? Molecular Transducers of Physical Activity Consortium. MoTrPAC is the study of exercise, okay? Obviously, we all know that exercise is important. We don't necessarily know why it's so great and how it gives us so many benefits. And so MoTrPAC is all about collecting multi-omic data in a trial of exercise. So I mentioned that we know that exercise is good. This is some proof for it. Maybe you didn't need it, but we have it. A little is good, a lot is better. And you can show that moderate or vigorous physical activity improve all-cause mortality and cardiovascular disease mortality. So it's clearly beneficial. Again, we don't totally know how.
So the Black was showing moderate's better than vigorous.
No, black is vigorous. So this is the hazard ratio. So these are a little better.
You die more.
This is worse.
Hazard ratio for
More people alive. More people alive. Yeah, no, good question. So the study goal is to study the multi-omic changes that occur during and after exercise and generate a molecular map of the effects of exercise and exercise training. Okay, so here's a map. This is actually all the sites that are involved. It's a huge consortium, many sites. There are clinical sites that enroll the patients. There are coordination sites that oversee the clinical trial. That includes us, the Bioinformatics Center here at Stanford. Then there's chemical analysis sites that actually produce the omic data. And there's actually it's also a rat study. So there's all the preclinical study sites which actually do experiments on rats and collect multi-omic samples. So this is an overview of MoTrPAC. Let's don't try to understand everything on the slide. Suffice it to say, MoTrPAC is multi-organism, so rats and humans. It is multi-modality of exercise, multiple durations of exercise, acute and chronic. There's multiple tissues. The rats get all their tissues collected. Humans still get 3, which is quite a lot, not just blood. We run it across, a dozen plus omic platforms to gather all of the data. And then this half of the slide is the Bioinformatics Center. It looks like the Bioinformatics Center is doing half the work. That's a misrepresentation. We are trying to contribute at all levels, but there is quite a lot that has to happen once that data is collected, and that's really where the BIC or the Bioinformatics Center steps in to take over. So let me give you a little bit of an overview of MoTrPAC, just so you can understand how it was at a basic level designed to be executed. So we take human sedentary individuals. So these are folks that are living their lives but not exercising. And we say, hey, do you want to join an exercise trial? We randomize them 2 to 2 to 1 to either resistance training, endurance training, or control. If they come in, they do an acute bout of exercise, whether it's endurance or resistance, over a course of about 40 minutes, or they rest if they're in the control group. Then they go off and they do exercise training for 12 weeks. And then they come back and they do that bout again. So we can learn how they respond to acute exercise. We can learn how they respond to training. And we can learn how their acute response changes after they've trained. All right, so we can do all this in this trial. And this started before COVID. And so unfortunately, the lightning bolt of COVID hit. These people had to basically stop coming to the trial. All of the clinical centers got shut down, which was really quite a bummer. But it means that we are going to talk today just about a subset of individuals who at least completed that first acute bout. This is where we have most of our data right now. It's 175 people who were randomized and completed the first acute bout of exercise prior to the COVID-19 shutdown. I'm really going to focus on this group. So who are these folks? They are predominantly female. Average BMI was about 27. The waist circumference was about 92. Their VO2 max, which is a measure of their endurance exercise capacity, was about 25. And I can tell you, if you're not familiar with these patients, these are overweight, sedentary individuals. These are definitely high numbers, right? So, and the VO2 in this case, that's a low number. Okay, so first we collect a bunch of data on them, a wide range of fitness testing, strength, endurance. We do some things called 1 repetition maximum, where you basically try to lift as much as you possibly can all at once. And all of this helps us understand how we should structure their exercise plan so that everybody is doing the same relative amount of exercise. That's really important, because if I work out 10 times harder than you, my multi-omic is going to change a lot more than you do if you're just sort of, kind of phoning it in when you're jogging on the treadmill. So we wanted to keep everybody really equal relative to their max. All right. So what are they actually doing? Endurance folks, they get a 40-minute bout at 65% of their max. So we test them, see how hard can you exercise, how far can your body actually push yourself, and then we back that down to 65% so you can actually do it for 40 minutes. The resistance group does 8 exercises. They do 3 sets of each exercise. They start with the upper body, they move to the lower body. That's very intentional. As I mentioned, we get muscle biopsies from the lower leg, and so, or from the leg, and so from the lower body. So we want those to have just been exercised right before we start to collect samples. And then we have the controls. These folks have the easiest job. They sit around in a chair and do nothing. And it's very important to have controls, and I'll talk about that a little bit at the time, but the controls are essential. Okay, let's talk about their day. So all participants arrive in the morning fasted. They have a baseline sample drawn on all 3 tissues, blood, muscle, and adipose. They begin their 40-minute exercise bout, and then participants in the control and endurance group have repeat collections of just blood during the intervention at 20 and 40 minutes. At 10 minutes after exercise, all 3 groups once again get a blood sample. This includes the resistance group. And then at 15 to 45, 3.5 to 4, and 24 hours, all 3 samples get collected from the participants. And then it's run across a number of omic profiles. Now, not every tissue gets every ohm. We'll talk a little bit about these decision makings towards the end of the talk, but you don't necessarily want to run everything on everyone all the time, but this is how it's sort of played out. Everybody gets transcriptomics and metabolomics. Not everybody gets phosphoproteomics or ATAC-seq, which is a epigenetic marker. Yeah.
What do you have to do to incentivize someone to do these repetitive muscle biopsies? I mean, who would sign up for this kind of
That's a
thing?
great question. Not me, but there are people who are committed to science, and we do incentivize monetarily for their time and difficulty. There is a modification to the protocol which has now made it such that people are randomized to a temporal profile. So instead of, say, getting 4 muscle biopsies over the course of the acute bout, they only get 2. They get the baseline one, and then they're randomized to either get the early, middle, or late, and that makes it a little bit easier. And I'll add that it's really important, this is sort of the control group, there's a great question to address that issue, is that we actually see a little bit of an inflammatory response in the control group who got all 4 biopsies. Their bodies are reacting a little bit to the repeated biopsies, so it's critical to have those control patients because then you can remove that signal. All right, so this is an example graph of how we kind of model the differential abundance in molecules. We use a mixed model for repeated measures. And we look at each of these levels. So the relative level of endurance is plotted at each time point for each type of exercise. So with endurance, we have here in orange, resistance is in green, and controls in purple. And the significance in the model at an FTR less than 5% is indicated by a black dot or a black circle on that point. So as you can see here, plasma lactate rises significantly in both exercise types and then returns to match control. So this is like a very standard, this is exactly what the data should look like. Lactate's a known molecule that responds to exercise. So when you run this model over thousands of features and all these time points, what you find is that there are thousands of features that are affected by exercise, most notably transcriptomics in muscle and blood, phosphoproteomics in adipose, and while the number is smaller, the percentage of metabolomic features in plasma affected by exercise is actually also really high. And that makes sense because all those metabolites are like lipids and fuel molecules that are going to provide the body with the energy it needs to do what it does. So it's not surprising that they're being flooded out into the bloodstream. And so when we use this data, we can start to answer some of the key questions that we have. So what are some of these key clinical questions? Let's go from a clinical perspective. Can we use the molecular response to exercise to improve disease treatment and prevention? What are the biomarkers of exercise effect? Are there pathways we can target for disease therapy? These are some of the big questions that MoTrPAC wants to answer. And I would argue that we kind of have to convert these to molecular questions before we can really answer these clinical questions. So what molecular responses occur in the body's tissues when we exercise? How are they similar? And how are they different? How do tissues coordinate their response to exercise? What molecules are the main drivers of exercise response and how are they in turn regulated? And how do molecular responses differ between people? So to understand how genes vary in their behavior across tissues, we looked at differential features that changed in all 3 tissues. So this is some work led by a postdoc at University of Michigan. One of the great things about a consortium is that you actually get to work with grad students and postdocs from all across the country and get a lot of collaborative experience, which is wonderful. So the number of overlapping features is actually surprisingly few if you look at across the tissues, and that's because adipose had a sort of a low number of changing features. But notably, MYC, a proto-oncogene that encodes a nuclear phosphoprotein, which plays a role in cell cycle progression, apoptosis, cellular transformation, all things that can be important in exercise, was seen to be differential in all 3 tissues, but with remarkably different trajectories in each. So in adipose, there's an early increase, also shows it later, and then in blood, you get this bimodal decreased response you can see here in the middle. So is my cursor showing up there? No?
Not yet.
There it is.
Yeah, there we go. So you can see in the middle here, you've got this bimodal sort of decrease here, which is very different than the other 2. Okay, next we wanted to look for these things called exerkines. So exerkines are molecules that originate in one of the tissues. In this case, we've got blood cells, we've got adipose, we've got muscle. And then they are released in protein form out into the bloodstream to signal that exercise is happening. And we're interested in these because if you can capture those pathways, you might be able to teach the whole body that exercise is happening. And that may be a way that the benefits of exercise are sort of spread around the body. And so we have the data to look at this because we have all these tissues, we have all these zones, and so we can look for things that are changing in the tissues and then end up in the bloodstream. And so we look to see in blood, muscle, and adipose whether any of those 2 features were changing. Blood has quite a large number of things that show up in blood cells and their protein form ends up in the blood. Muscle has a good number as well. There's actually some decent overlap where both tissues are pushing things out into the system, or at least we think that they are. And we looked across all these tissues. We used an FTR cutoff of 0.1. And one of the most interesting findings, we found some known exerkines, but then we also found this novel one called CCN1. And so CCN1 is a secreted protein that's growth factor inducible. And it plays a role in cell differentiation, proliferation, angiogenesis, and then extracellular matrix formation. And these are really key pathways that are important in exercise response. And so CCN1 is, first of all, it's known to be a secreted protein. Variants in that protein are actually also linked to plasma triglyceride levels, which is a really important clinical metric. because circulating lipid levels can lead to cardiovascular disease. So we think that this is potentially a really important protein. And what you can see is that expression of this gene goes up with exercise in adipose and muscle. Actually, the protein levels go up in muscle too, if you measure direct the proteomics. And then the proteomics of the plasma show that it's flooding out into the bloodstream during exercise. And so we think this is a really novel candidate exerkine that we're going to be studying a little bit more. Interestingly, if you look at transcriptomic data from GTEx, so this library of transcriptomics, it turns out that you can use some tools to understand the correlations between expression of certain genes and the overall expression patterns of full tissues, which would suggest that that molecule is then signaling to those tissues to kind of affect their global transcription if they're correlated with one another. The top two tissues in that analysis that are associated with levels of CCN1 in muscle are muscle and adipose. And that's using all the tissues. So we do really think that this molecule is likely signaling to these two tissues. And so we're trying to study that a little bit more in the data that we have. Okay, let's switch over to a method here that uses a network of our existing features. And this is called SCION, spatiotemporal clustering and inference of omics networks. And I'm not going to get too deeply into the methods. I want to talk about it at a high level, but we obviously want to understand how these molecules correlate with one another. But one of the things that I really like about SCION is a networking algorithm that allows you to inject pre-existing biochemical knowledge or biomedical knowledge into it. And so you can start with regulators and then identify targets a priori, and then use the network to organize around those things that you identify. So we know that circulating metabolites and differentially phosphorylated transcription factors are kind of the drivers of downstream biology in any given tissue, right? Molecules circulating in the blood can come signal to the muscle, and then they activate perhaps transcription factors by phosphorylation. That's how they get activated, and then they have downstream effects. And so we can identify those groups as really important, and then we can ask the network to organize around those. And what you get is you cluster the data and then you organize the networks around them, and that's exactly what we did. And if you take one of these clusters, for example, you get as one of your hubs in the cluster NR4A1. This is a transcription factor that is already known to be important in exercise. So we like the path that we're on here. But then you can actually see one of the bigger hubs here is this protein TFAB, or transcription factor EB. Transcription factor EB is interesting because it's actually been shown in mouse models to be important in mitochondrial formation and autophagy. That's being targeted by some drugs, in fact, for those roles. Those things are very important in exercise because the mitochondria are the energy powerhouse of the cell. And so what we think is that TFAB may be really important in garnering some of those benefits, and it really seems to be a central driver when you integrate this knowledge. The great thing about multi-omic studies is that when you find something you're interested in, you've already done a bunch more follow-up experiments. So we can look and exactly see what proteins what residues in the protein are phosphorylated differentially. You can go straight to that data. You can then go to the transcriptomic data, and you can see that TFAB is then down-regulated late. So you're already starting to put together a temporally patterned picture because you collected all that data at once. And it's a nice way to be able to do, again, several experiments and explore your results. Okay, one more thing that I think is exciting to do with this data. So to understand heterogeneity of response to exercise, we identified molecular features for which the exercise response had a significant interaction with participants' baseline VO2. So to visualize these effects, we show here the response of 4 plasma metabolites grouped into quartiles of their baseline VO2 max. So we basically identified those that are not very good at exercise and those that are much better at exercise. And we look at how their metabolites trend over time. And as you can see, in a significant way, these 4 metabolites show very significant differences between whether you're fit at the beginning or less fit at the beginning. And this is a good sign because this means that we can use our multi-omic data to try to understand different responses in exercise among individuals. And that may allow us to then one day, and this is a longer term goal, but to actually try to tailor your exercise to you, because we're seeing that your multi-omic is really responding differently. Okay, so a little bit of conclusions of this section. So what molecular responses occur in the body's tissues when we exercise? How are they similar? How are they different? We found that MYC expression during exercise is highly variable across tissues and time. How do tissues coordinate their response to exercise? CCN1 is a candidate exerkine from muscle or adipose tissue. molecules are main drivers of exercise response, and how are they in turn regulated? We see that TFEB is a hub of circulating metabolites and muscle transcripts and is differentially regulated in exercise. And then how do molecular responses differ between people? We see that baseline fitness is defined by beta-2 max leads to graded metabolite responses during exercise. Okay, how is this all going to help Mr. Jackson and Mr. Ahrens? And I would argue that we are not there yet, but we're on the path. And I want to talk a little bit about where that path is leading. So why is exercise, we know why it's good for most people, but why is it specifically good for heart failure patients, the patients that I take care of? Well, this is a study that was done in 2009 called HF Action, and it basically sent people off to cardiac rehab who had heart failure. And what it found was that after cardiac rehabilitation, the most important thing that changed for these individuals was their quality of life. They felt better. And I know these curves don't look that different, but actually the percentages of people who had a meaningful shift in their quality of life score was about twice as high, 59% versus I think about 23% in the non-intervention group. And so this is what really said, you should send your heart failure patients off to do exercise. But again, why is exercise, it comes back to that original why question. Why is it actually good? What is happening to these heart failure patients that's making it different? It's because I'll tell you, their hearts are not getting better. If you or I were to start to go exercising and we have normal hearts, our hearts will adapt to the exercise. They will get stronger, they will be more efficient. Heart failure patients' hearts cannot do that. Their hearts are broken. And so unfortunately, they have to adapt in other ways. And my hypothesis is that there's a real role here for vascular adaptation. Something peripherally is going on that is allowing these patients to do more. And so based on prior data, we think that vascular extracellular matrix proteins are really markers of vascular adaptation. We want to try to study those better. Why do we think that? Well, we're back in the land of volcanoes. When we did a study looking at the proteomics of exercise training in a separate cohort, we saw that these vascular VCM proteins were linked to the amount of improvement in people's exercise capacity. And so the thought is that these proteins may be playing a unique role in heart failure because we know that they are abnormally regulated in heart failure as well. So we want to try to look at this, which brings us to a trial that we're presently running where we are trying to mimic MoTrPAC, but in heart failure patients. And we're using some of the same platforms, some of the same protocols. And what's great is that we'll have MoTrPAC to compare to. So we'll be able to say, that's the whole point of MoTrPAC, is to be this map. We'll be able to say, we have heart failure patients that are having these multi-omic changes en masse that are different or the same as those that are healthy. Okay, let's talk about another reason that I think this is going to be good for Mr. Ahrens and Mr. Jackson, which is we have also other data in MoTrPAC that might be more immediately relevant. So the RAD data, which I haven't talked about yet, right now it's just in chronic exercise, but we collected samples at 1, 2, 4, and 8 weeks of endurance training in rats, and then compared that to control. And as I mentioned in rats, you can get all the tissues. So that includes heart, as well as many others. One of the things that's been most amazing about this study is the fact that every single tissue responds to exercise on a multi-omic level. You can see that every tissue is affected because in this matrix, you can see the number of changing features across the omes on the y-axis and across all the tissues on the x-axis. So let's dive into heart. So this is work that's being led by one of my postdocs in the group, Pauline Brochet, who has worked previously in France on multi-omics in Chagas disease. So she took on this data set of the heart multi-omic data and said, okay, let's integrate all of this data. We have a lot of information across, this tissue across, 4 different omic categories, but 9 different platforms. Let's dive into this. And she utilized multi-omics factor analysis, specifically the Mephisto algorithm, which is really good for this temporal data because we're collecting data over time. And so it creates these smooth changes in factors over time. I'm not going to get into the math here, but I'll go jump here to the results, which is that it identified 4 uncorrelated factors within the entirety of the multi-omic data. And I think what's great is that you can see there are some unique behaviors here. You've got factor 2, which is this continuous up. You've got factor 3 and 4, which is sort of this immediate early up at 1 and 2 weeks, and then a downtrend back to baseline, early up at 1 and 2 weeks, and then down to baseline. And I think, in maybe in almost an anthropomorphized way, you can sort of think of these as, here's the long-term adaptation, the things that mean these animals are really different after the exercise training. And these are the things that are the machinery to affect that change that only need to exist for as long as they're initially adapting, and then they go back to baseline. So we have the adaptation and we have the long-term change. And it's important to see that the long-term change, which is summarized sort of across the multi-omic, is actually associated with the change in VO2 max of these animals over time. So you can see that the changes that we're getting cardiovascularly as measured by VO2 max are correlating with this factor. And so we think it's particularly important. We wanted to understand how these factors, what was making up the factors, what was driving these changes at a molecular level. And so we do pathway analysis to figure this out. I'm going to actually show factor 3 here. So factor 3 is that early on and then back off that seems to be earlier in males. And you can see here that if you look across all of these ohms and in these various pathway categories in factor 3, that there's this upregulation of fat digestion, fatty acid elongation, aerobic respiration. What it tells me is that there's a change in the energy metabolism in these rat hearts. A little earlier in the males, a little later in the females, that sexual dimorphism is important, but that's happening, and that may be an adaptive element of this, and that really tells us more about that cardiovascular biology. We're looking now to see whether these patterns are actually the opposite of diseased heart failure hearts. Towards that end, in order to better understand the interplay between exercise effects and cardiovascular disease genes within the heart, we started with a set of genes clinically known to play a role in cardiovascular disease, and then we looked specifically at their behavior in any omic layer over the duration of exercise, training in male and female rats separately. So as you can see, many of these features, you've got all the features kind of lined up here on the rows. You've got the evolution over time on this axis here. And you can see that in the females and the males, there are sort of linearly increasing or linearly decreasing levels of the omic features that correspond to these cardiovascular disease genes. And so just as an example, I'll highlight the CRY-B protein here, which is upregulated at the protein level and at the phosphorylation level. This protein is known to have a critical role in reducing pressure overload induced cardiac hypertrophy. So the multi-omic data has led us to the importance of this particular gene. And the hope is that, if exercise is really sort of improving and increasing levels, that probably it's not just that you need to have a mutation in this gene to have a problem. You could actually increase the levels of it and it could do more and get more benefit. So I think there's a couple of ways that we're going to start to go down the path of helping Mr. Jackson and Mr. Ahrens. So in the last bit here, we've got about 20 minutes remaining. I want to talk a little bit about the Bioinformatics Center, and this is a great group to do it in because I think a lot of people here either deal with or struggle with, as may be the case, some of the same issues that we deal with in the Bioinformatics Center, handling all this incredibly rich and diverse multi-omic data. And so I wanted to talk a little bit about some of our experiences, some of our I think triumphs and some of our struggles, and see what people's thoughts are, because I know that everybody is sort of in this room deals with these as well. So this is the MoTrPAC data hub. It is a website. You can click that. You can go through that QR link. It's not a bad one, I promise. And you can go to the website and explore our data. There's ability to download the data. There are video tutorials on how to use the data and how to use the website. There are ways to explore the data without downloading it. You can just—
Is it public?
Yeah, this is all
The
public.
data's public?
Yeah, just the chronic rat data. But as we generate more, it will get increasingly public. So yeah, you can start to work with these data sets right away. So for example, the cardiac data that we're working with, that is public data. You could work with it as well. And so I want to talk a little bit about the importance of tools and browsers like this, and I'll do it by highlighting a little story really quickly. So I'll go back to the GWAS pipeline that we built, which we then put together into a tool that we were using internally at the time that we nicknamed the database of multi-omics, or DOMO. And during the pandemic, on June 2nd, 2020, which was at the height of the pandemic, this paper came out in preprint and then eventually came out in the New England Journal that linked variation in blood type to COVID-19 severity. Got a lot of press, if you guys remember. And that came out on June 2nd, and then on June 3rd, the New York Times picked up on this, and they interviewed the lead author, and they asked him, so why does blood type affect your response to COVID-19? And he said, I don't know. And he said, that is haunting me, quite honestly. So the next day, I went to our database and I looked up the variant that they found, and I found that the most strongly associated protein with that variant was CD209. And when I looked up what CD209 does, it is a binding site for viruses. It is known to actually bind SARS-CoV-1. And so even though the ABO gene, which is where the variant was, it obviously makes the ABO enzyme, that protein, and so it's associated, of course, with the levels of that, but it's also associated with the levels of CD209, which is this binding protein. So we really think that that's probably why this was occurring, is because there were changes in the ABO gene were linked to then changes in this binding site for viruses. And so we put out a paper 9 days later from the day that the original preprint dropped, that eventually got added as a letter. But I think it highlights, it's a cute story, and COVID is over now, I know, but it highlights how important it is to have this data at the ready in tools that people can use without having to code it up, because people may need to make these insights in a rapid way. And so we're trying to develop this now to make it more broadly available. This is sort of the newest iteration of it. But I think it really highlights why I think it's so important to, do the work of the Bioinformatics Center and to make this data easy to use and easily accessible. And so I kind of organize the layers of the BIC into 4 places. It's organization and collaboration, it's data ingestion and processing, it's analysis, but then of course critically it's dissemination. And so the center has this is sort of focusing on data ingestion and processing, has really tried to innovate at every layer of that process and tried to make the process either easier, better, more efficient, faster, less resources. So for example, obviously we, as part of the organizational process, we work with all of our data producers to understand their data, understand their needs in terms of getting the data to us and how much processing it needs after the data is transferred. We then create this environment in a system that's been developed at the BIC called Omics Pipelines, which is a encapsulated area where we run all of our genomic and proteomic pipelines. It's a proprietary tool that was developed in the BIC, led by Mihir Samdarshi, who created this in order to be sort of a really diverse tool for self-hosted and valid, easy to use, private and secure way of handling all of your omics pipelines. It's really easy to use and it's in full swing at the BIC now. And on top of that, if you go to the website and log in, so this is not public, you notice I've logged in at the top here, this is for internal users, you can go to the QC data monitor and any of the data producers can get a live dashboard of how their data is progressing through the pipeline. Has it been fully processed? Is it having QC failures or warnings? And they can explore this so that they're getting a live look at how their data is progressing. This has made it a lot easier to say, to take a little bit of human interaction out of the loop, save us a few Zoom meetings, because people know where their issues are in the QC process. Okay, let's talk about analysis. The same thing. I think we've tried to make the analysis process a little bit easier and a little bit more democratized across our consortium. In our consortium, there are a lot of data people, mostly at the BIC, but at all the sites, they know their data really well. But not everybody is, ready to sit down at the command line or ready to code something up in R. And we want to make sure that they have, they have great molecular, biological, and physiologic insight. We do not want to make this data only for the coders right out of the gate, because they actually need to do some feedback. So if you log into the website, there's also this link here called pre-COVID human data visualization. So this data is not out yet, but we have created, before the data is even ready to be released, a summary of a lot of the early analyses that we've already run on the data. So internal users can go to this dashboard, they can type in their favorite gene, and they can browse in an interactive way on the most up-to-date results as the analysis is progressing. It's sort of like building the plane as you're flying it, because we're running these analyses, we're generating this data, and we're trying to immediately have it available, not just to the analysts and the coders, but to have it immediately available internally. And then, of course, the communication is critical, and shared environments are critical. We try to maintain shared cloud environments for all of our users, really focus on security to make sure that people are validated before they go into this. And especially, it's still in a pre-release environment. We haven't released the data publicly, so we're all cleared to work with the data, but you still have to worry about security at that level. We try to create shared code repositories. And particularly, we try to create shared functional packages that allow all of our analysts who are working on the data simultaneously, often across multiple OMs and across multiple processes, to try to save time. Again, I'll use that analogy of building the plane as you're flying it. As people are developing functions and visualizations of the data, we're then feeding that back into the package so that anybody can quickly generate that for their tissue or their OM of interest. and gain the insights, even though, let's say I was working on blood metabolomics, I generate a cool visualization, you were gonna wanna generate it from muscle proteomics without having to redo all my work. So we're constantly trying to create that environment at the BIC, which is a real challenge, I have to tell you. It requires a fair number of meetings and a lot of data pushes. Then let's talk about external release. So I'll just highlight the data hub because I think this is one of the most important places of the release. This is public. So this is ways of browsing the data. So all those visualizations that we built during the analysis phase, we flip them to external, and now anybody can browse the data using these tools and start to gain insights about the trajectory of molecules and exercise. And so this is fully interactive. You can customize the visualization. There's a lot of different plots that you can build on the website. And you can download tables of data and you can generate figures and potentially use those figures for your grants, for your papers. We actually brought this up at a conference not too long ago on exercise, and there was like a little bit of panic in the room of like an idea that I could access the same data as my colleague over there and we could end up putting it into a paper independently. Won't that be a problem? If she's using it and I'm using it, and I said, I think that's actually the goal. We want you to be using this. Obviously, we want you to cite, we want you to reference, but that's better to us than this data sitting behind a firewall and not being used. I would rather you both publish something and come to the same conclusion. But it was a little bit for sort of some less data inclined people, that was a bit of a shock that people could be looking at the same data.
Professors.
I was like, what'd you do?
So looking at the bioinformatics side, we try now from a high degree, we try to be the fairest of them all. Obviously, we can always be more findable, accessible, interoperable, and reusable. And so we're trying to be as fair as we can. And across multiple domains, we're trying to get people to interact with the data. So we try to communicate as well as we can. We have a discourse set up. We have office hours so people understand how to access the data. We've got source code that people can go use so that they can reproduce all of our analyses. We make packages to make it easy for people to get up and started with the data. You can go download the chronic exercise data from the rats and work with it in a package. Or you can get it from our cloud website and download it directly there. Or you can go to one of the external repositories and find it there. We want to try to get data into people's hands wherever they are. And so it means that it has to be released in a lot of different places. All right, so I want to talk a little bit about challenges and visions for the future. And then I'm excited to hear everybody's thoughts and feedback, particularly about these things, because these are the things that I feel like I struggle with every day. So multi-omic data application requires domain, technical, biological, computational, and informatics expertise. You really need the right team. In an ideal world, I feel like it's best if certain members of the team have multiple areas of expertise across these things. And because I think, you spend a lot of time trying to understand unique elements of the platforms. Remember, these are your data is only as good as your measurements. All those measurements come at a cost. This is sort of an overview of how much all these ohms cost. A single ohm for a multi-ohm for a single person can be like $2,000 or more. And so you want to make sure that you're getting the most accurate and valuable data. Oftentimes specificity, cost, and throughput in these platforms are at odds with one another. And so we've studied this pretty in depth about these two proteomic platforms. These are just two of the many, but we've done extensive comparisons of these two to try to understand their strengths and their weaknesses. It's that kind of comparison that lets us get better data, but it's something that's really critically important. So I think about this: can we make the process faster, more adaptable, less human intensive? Despite the innovations that I hope we've made to make the process better, I think from data generation to data release is still taking like 2 years. It's way too long. It's way too. Could we get that to 2 weeks? Can we make all of these things more automated by leveraging, whether it's artificial intelligence, by developing pipelines that make this go faster? In particular, I think by having better metadata, we can definitely improve the speed there. How do we learn faster from multi-omic data? So this is from a recent review that we did, highlighting a lot of multi-omic studies and exercise. Every one of these dots is an exercise. MoTrPAC's down in that corner. It's the biggest and it has the most data, but it does not talk to all of these other studies. You cannot take the data from one of those big ones up in the left-hand corner and see if it matches up with what we're finding in MoTrPAC in a readily accessible way. That is a problem. And I feel like we are trying to address that. I think many people are trying to address that because it's really important to get these data to talk to each other. And so I think critically important to improve the data and metadata standards so that data can readily connect and be AI and ML ready. We talk about this a lot in the Common Fund, which is where, MoTrPAC is part of this NIH group called the Common Fund. There are other studies in there. We are trying to do a project right now with the LINKS Consortium. It's just 2 data sets. And the difficulty in getting those 2 data sets to integrate, even despite the fact that LINKS has created incredible tools to help with this, it's still challenging. It's still time consuming. So we want to try to address that. Continued emphasis on controlled work environments and data security. Right now, at least for internal work, we're still kind of working, sometimes on, they're institutional computers, but they're on-prem. We'd love for this to be all cloud-based. We'd love to be able to quickly set up environments so that people can go to the data rather than the other way around. But we have to do that in a secure fashion, and that's something that we're still working on. And then integration into the electronic health record for research and clinical purposes. I think this is something that I know people in this room would be excited about, because I really think that as the multi-omic data as I hope I've demonstrated, it's useful, but we're eventually going to get to the point where it can be useful on an individual level. Knowing one person's multi-omic can do a lot of things. It might be able to identify a diagnosis early. It might be able to identify them for a clinical trial as a particular, subtype of a heterogeneous syndrome, as I identified. It might allow them to get the treatment sooner. So I think we want to try to get these data increasingly linked to individuals. We're doing a lot of multi-omic studies here at Stanford. There's no reason that you can't back. So with that, I'd love to hear people's thoughts about those issues or anything else. A lot of great people at the BIC that I'm really privileged to work with. A lot of great people at MoTrPAC and our labs. Thank you for your time and attention.
After questions, follow-ups?
So I have a comment.
Yes.
As long as people are worried about data, the same people using, different people using same data,
Yeah.
you won't get to have your process that releases data in 2 weeks. That fundamentally has to
I know. It's scary to people. In TOPMed, which is, again, this is that consortium of all of the cardiovascular studies, it's even worse because these are data that the cohorts feel some ownership over that data. They take a lot of time and energy to generate it, and to just sort of release it to the world feels scary to people. And yeah, I'm sure you've done that with a lot of TOPMed cohorts, Framingham. It's just hard to get in there. It's a shame because it can do a lot of great stuff, and maybe a policy level thing,
Yeah, I was gonna say, so at the NIH level, they're trying to associate, not like a PubMed ID, but something like that. They've been talking about it for so long, to data sets, so that just like you get credit for publications, a number of citations, you would equally get credit for data use and things like that, so.
We even tried, I was new faculty member, I think that was like a 2nd or 3rd paper as a new faculty member. And New England Journal of Medicine article had come out with research parasite editorial. So for that paper, we actually added every author that we used the data from. And the editor balked and said, nope, we are not allowed to.
It's everything, it's journals, policy, I mean, it's a huge shift.
The incentive structure is not set up to help people do that, despite the talk about it. You've, I'm sure, heard. The incentives just aren't there, and particularly some of these cohorts. I mean, people working as a fellow in those cohorts, they can make their whole career on those data sets, and doesn't feel that great to give up the direct access.
But they should be able to make their career. They don't have to give that up, right? I mean, and that's where we have a hard time.
But I mean, there is a problem, right?
Yeah.
If you are going to make your career on just that, you are thinking too small, right? Like somebody should be telling them that like, there is a lot more you can do. Don't focus on just this one thing.
Squirrels will hoard acorns, come on.
It's a
I'm
human
trying to
instinct.
get the conversation back to your work.
Oh, no, it's okay.
So 2 things. One, you should set up a booth and do muscle biopsies on all of us whenever we.
Yeah.
I'm just-
See, I'll get my biopsy. We'll do it now.
Do it after the annual review.
Really inflamed. What a signal here.
You create this incredible picture of MoTrPAC, a cast of thousands working on all this stuff. People in this room want to know how to collaborate with you, perhaps.
Yeah.
So maybe we're spending a moment just talking about where you see your own work going. and
Yeah.
what you would be doing that people might be able to join in with.
Absolutely, absolutely. So I think a lot of it comes from these questions, the talk that I want to give in 5 is probably wrong, 10 years. The talk I want to give in 10 years is to have a lot of answers to these questions, to figure out how we answer these things. How did we get the data to process through a clinical trial faster? And so what we're going to start working on, and what I hope people would want to work with me on, is systems that allow us to do things. So think about all of the speed bumps along the way that we could address as a group working together. So we could improve the ability for people to get data into the system. There's no reason that we can't leverage AI to take a data set that is not in the right structure and get it into the right structure without us having to go back and forth on 6 Zoom calls to say, actually, you need to move column 2 to column 4 and whatever it may be. The omics pipelines is a great start, but we need to capture sort of a more limited set of objects so that we can oversee those pipelines in a way that means that we know when things break and we can go back and fix them. Those breakages happen a lot, and in the cloud level, we don't always detect them early enough. So we're developing omics pipelines, which is really just an entirely computational architecture. So if you're interested in that, that's a place to do it. When we talk about making data talk to each other, we need a way of utilizing the data that we have, probably through large language models, to generate omic data at an organized level that can then be connected to other omic data at a global level. So, for example, I mentioned this graph. So all of these studies are published. Their data, at least in what's available in supplemental tables, et cetera, is available. We need to start to gather that data. I'm hesitant to say sweep, but we need to start to gather that data in an automated way to try to integrate it all together. So there's another avenue of data integration at that level. So I think those are some of the things that we.
So I'm just gonna push on Mark's question. So I mean, we're a fairly small group, so you know what we do. So I heard you mention knowledge graphs. I mean, like, is there opportunities to like work with Mark on knowledge graphs? I'm sure you've been talking with, I've heard you've already been talking with Pravesh, so that's it. But like with Nigam about how you might sort of get some of this in the clinic or with some of these other, I mean, so given we have a small expertise, I mean, I would love to know like how you're integrating knowledge graphs in there. Have you thought about that? Is that something you could think about?
Yeah, I think absolutely. So Mark and I met actually to talk a little bit about the metadata issue, because that is a key part of getting these data sets to talk to each other. So Common Fund, which has a lot of multi-omic studies in it. It has a data model, and it has the rules for their metadata, and it stops at the level of multi-omic. So like you have a multi-omic matrix, say, of all the measurements, and it says just say where the matrix is, but it doesn't organize the data within that matrix. It doesn't give you standards for that. So we need to work on that next layer, and we've had conversations about that. I think I'm working to get a meeting with Nigam to talk a little bit more about integrating multi-omics at the EHR level. We have more work to do there, because I think that there are problems with interpreting a single person's multi-omic, but nonetheless, we still want to do it at a more global cohort level. And I think I've had conversations with you in a little bit about that. I think he's interested. Obviously, his involvement in this trial is important too. So, and then in terms of knowledge graphs, this is something that we're just starting to scratch the surface on a little bit, but I think that that's one where I want to do a lot of development. We've envisioned this concept of the Xer hub, where you should be able to do. The tagline for it is stop Googling genes, because when we found CCN1, the first thing we did was Google it. And that seems wrong, right? Like we have much more higher level knowledge data that we should be able to integrate into a knowledge graph. The Common Fund is doing some of that and wants to use our data to do that. It's not going great. And so I think, just to be really honest, and I think that we have an opportunity to do better. So yeah, all those things are possible.
Fantastic. We are at the hour. Thanks again to our newest faculty, Dr. Katz. We have one online question about this. I guess is talk to each other. I think you were kind of naturally answering that already, so we'll follow up. Yeah. Thanks, everybody.